larger canopies, may get caught up in the surrounding branches of the foliage as they're descending. And as they decay through their descent, their decomposition will supply other transitional ecosystems with the nutrients that they need for their productivity before reaching the ground floor, limiting the amount of nutrients available to that ecosystem.
I used to like that bigger loop, seeing the leaves fall, and the nutrients going into the soil, and then going back up. But what we were kind of looking at is whether there's prior decomposition that takes place before the leaf actually hits the ground, because it gets caught in another tree or something else. What we did is we chose 3 trees, all almond trees. One was tall, one was very tall, and one was extremely tall. And great descriptions, the heights are in the data. But we did a pilot study. So instead of using true replicates, we only did pseudo-replicates for the limitation of time. So what we did for our tall tree, instead of picking 5 tall trees, we picked 1 tall tree and we did 5 leaves from that same tree, and on and on to the control, and 5 leaves that were experimental. And what we did here is we had part of a card, we cut out 3 by 6 centimeters, and for each of our leaves, our control and our experimental, we found an area on the leaf. The leaf is asymmetrical, so what we did is we tried to aim about the middle of each leaf and use a scalpel to cut out a large area of the leaves. Control, experimental, those leaves were placed in 1 petri dish. So 5 leaves from the control, those cutouts were placed in 1 petri dish. Okay, 5 cutouts from experimental in 1 petri dish. Okay. All right. And so what we did was we went to the drying oven, put it on 40 degrees Celsius, made sure the temperature was constant. We kept the petri dishes, which were open, so the leaves were exposed, for 24 hours to dry out all of the leaves. Okay, and then after 24 hours, we weighed each leaf.
Data analysis, I think most of the groups probably did. We did collect tree height and tree girth. In the end, we decided to use tree height as our primary indicators because that was directly connected to our hypothesis. We calculated the mean dry biomass, so we dried them in the ovens until there was pretty much very little moisture left, so we could get just the carbon and other materials in there. One of the key things here is we calculated percentage of decomposition. So this is how much of the mass